I'll do it again. Advanced question screen share. Stop.
जवेरे जल्दी आओ.
Right. So this is an intestinal, when it comes to intestinal protozoa, you have quite a lot of unicellular eukaryotic organisms basically. So when it comes to amoeba, the spelling is wrong over here. You should write amoeba, Amoeba. So it was formerly in the phylum Sarcodina, in fact, but right now that phylum Sarcodina is actually class Sarcodina. So phylum Protozoa, subphylum Plasmodroma, class Sarcodina, order Amoeba or Amoebina or Lobosa, genus Entamoeba, species is Histologica. So as you get it. So it's one of the most pathogenic enteric. Enteric means in the intestinals basically. Parasites which causes amoebic dysentery in man, or it causes amoebiasis. I request you all to please mute yourselves. Please mute yourselves. Thank you. Right. So this parasite is mostly prevalent in Mexico, India, China, parts of South America, and wherever there is unhygienic sanitary conditions which are present. So it was first discovered by Lambie in year 1859, in fact, and the parasitic nature was discovered by a Russian zoologist by name Losh, and it lives in the lower portion of the small intestines. It lives in the mucosal and the submucosal layer of the entire large intestine, that is the colon. It also occurs in the appendix, and it is a a monogenic parasite. Monogenetic parasite, meaning its entire life history takes place in only one host, that is man. So some species are parasitic, some are endocommensals. Again, they're found in cohabitant with different animals again. You can find entamoeba or endocommensals in domestic animals, birds, reptiles, frogs, you name it, they're there. Even in Hydra, which is a coelenterate. So the pathogenic ones cause amoebiasis. Easis means a pathological condition. So when it comes to Entamoeba histolytica, it's one of the most important parasites that's present. So dogs, cats, primates can also be infected, but they're quite rare. But it's primarily a human parasite. What about guinea pigs? No, not there in guinea pigs, as far as I know. Chalo. It's transmitted from human to human also. So if you see, it was first seen in 1878, but it had not been described till 1903. Then it causes amoebic dysentery, and this is the picture of Entamoeba histolytica. As you can see, this is a pseudopodium, slight one which can come out, and there's a food vacuole along with RBCs in it. See, then you have the nucleus. This is the endosome, and then you have a that's what food vacuole with, and these are the chromatid or bar bodies. So the adult parasite is called as a trophozoite, which is the harmful stage basically. It's about 18 to 40 microns, in fact. See, it differs according to whatever they kind of gave over here in this PowerPoint. It says 20 to 30 micrometers, but it's usually 18 to 40 microns. So follow your book, please. For whatever reason, please follow your book. The body is with a semi-permeable plasma membrane or plasma lemma. Cytoplasm is differentiated into ectoplasm and inner granular endoplasm. And when it comes to that, nucleus is small, round. Bacteria are found inside the endoplasm basically, and you also have a delicate ring of chromatin in the periphery. As you can see, you have the chromatid bodies as well, and then many food vacuoles which have ingested RBCs in them, and bacteria are found in the endoplasm, and they have a clear hyaline, blunt, retractile pseudopodium which is present, and this makes the what do you call it, entamoeba to move in one direction only. Contractile vacuoles are absent because this entamoeba histolytica lives in an isotonic medium wherein body fluid concentration and the entamoeba histolytica concentration, whatever is there, is the same. So what happens is you do not need a contractile vacuole. As such, water does not enter through osmosis in the case of entamoeba histolytica. If at all you see the life cycle of it, as given in your textbook, please follow the textbook picture. There
Ma'am,
it's got a good what do you call it thing. I could not get a new picture actually. Let me see if at all I can do it.
ma'am, osmosis. What is osmosis?
Osmosis is water traveling from a concentration of a higher concentration to lower concentration.
Okay.
Okay, so what happens is if the concentration is different in the medium, for example, the blood concentration of blood is different from the concentration of the organism. So what happens is there is a traveling of water. So from the solute, what do you call it, molecules of a solvent, they through a semi-permeable membrane. For example, over here it is basically a plasma lemma only. So from a more concentrated solution, the solute or the molecules of solvent will be traveling to the what do you call it, less concentrated. I'll show you a picture. So what is osmosis? Right. Let me share this if possible. Are you able to see? Acha, it's resume share. So if you can see, this is the solvent molecule. This is the solute molecules which are present basically. So the concentration is more here, the concentration is less. So what will happen is, finally, you get to see that, maybe this is not the right picture. Let me see if I can get a better picture. Right. So basic difference between osmosis and diffusion, if you come to it. Please wait a minute. Right. So in osmosis, there's a semi-permeable membrane through which the solution water goes from the region of higher concentration to lower concentration, or the solute concentration is more, solvent concentration is less, because of which water travels from the lower concentration of water to higher concentration of solutes. Is this clear for you?
Yes, yes.
Cool. Chalo,
Yes, ma'am.
Let's go back to, so anyway, this is not going to happen in entamoeba histolytica because the concentration or this entamoeba histolytica lives in an isotonic medium as such. So what happens is there is no movement of water from the host, that is man, to entamoeba itself. Sorry. I'm trying to get a good picture of entamoeba histolytical life cycle basically. Let us see if we get one good picture. Let us see if this is good. Is it? So this is the picture as given by your CDC, that is your, if you can see, anyway. So these are the mature sister trophozoites that are present. You have a mature trophozoite which undergoes binary fission. After undergoing binary fission, you have a young trophozoite, or just follow whatever is given in your textbook. That is a beautiful picture again. You have a uninucleate cyst, you have a binucleate cyst, you have a tetranucleate cyst also that is being formed. And this tetranucleate cyst emerges out. I have that good picture actually, if possible. So uninucleate, binucleate, and tetranucleate cysts. That's coming over here. And then the tetranucleate forms will keep coming out or emerging as the infective stages, which is the metacystic trophozoite stage. And this is how locomotion takes place. Basically, as you can see, this is the amoeboid movement. So there is actin and what do you call it? Cell membrane, which is, the sol gel theory, as some of you might have heard or learnt about. So that causes the amoeboid movement basically. So low and high osmotic concentration inside the plasma lemma or ectoplasm and endoplasm causes the formation of the pseudopodium. Anyway, as given in your like what do you call it, by the CDC, Center for Disease Control. So the infected stages are the metacysts basically, or the and the trophozoites. So the trophozoites also have ingested RBC, nucleus, endoplasm, ectoplasm, the plasma lemma, whatever is there, cyst walls, glycogens, chromatid bodies, all of them. And now there are 4 stages in this parasitic life cycle, which is the trophozoite stage, the precystic stage, the cystic, and the metacystic. So this is all about the life cycle basically. So when the cyst gets ingested, existation takes place in the small intestine. You get so this process, encystment is the process wherein transformation of trophozoites to cysts takes place. So after feeding, growing by multiple, by binary fission, basically, trophozoite comes out of the tissues into the lumen of the large intestine. And what do you call it? Once they go into the large intestines, what happens is the parasite becomes spherical, small, food vacuoles disappear. Then these are called as the precystic or the minuta forms. I'm trying to see whether I have that picture with me. I guess I don't exactly have that. So I'll just stick to this picture, whatever is there right now. Right. So the single nucleus non-granular cytoplasm form, whatever is there, is actually a uninucleate precystic form. And and then once this is formed, it divides 2 times by mitosis to form 8 into 4 daughter nuclei. Then it becomes a tetranucleate or the quadrinucleate. So tetranucleate or the quadrinucleate. And then what happens is, once this is done, you have the quadrinucleate, which is around 5 to 10 microns in diameter. And this stage you have chromatid bodies which are large and you can see that this is the characteristic feature of the cysts of entamoeba. Now, the complete process of the encystment to existment or encystation itself is taking place only in a few hours. Now, the cysts are the infective stage of the parasite, and they cannot resist unfavorable conditions, be it very high temperatures or be it there is no water or extremely high salinity for that matter, or all such kind of unfavorable conditions kill the cysts. But then once the cysts are anyway there, the mature cysts pass out intermittently through the feces of the man. Then they remain viable for a long time outside the human parasite. Then what happens is, if you consume unhygienic or contaminated food or water which is contaminated with this Entamoeba histologica, this causes new infection in a new person as such. So contamination can also be occurring through house lice, cockroaches, and other unhygienic food handling, etc. Now, when it comes to existment, as you can see, existment causes the cyst to form trophozoites. Excuse me. Right. So this occurs in the formation of the trophozoites occurs in the alimentary canal inside the man basically. So when the food contaminated food reaches the human intestine, cyst wall gets dissolved by digestive juices. Tetranucleate stage, whatever is there, comes out. This process is known as existment or metacystic stage. Now, metacystic undergoes a series of mitotic divisions, forms 8 multi-uninucleate daughter amoebulae. Now, these daughter amoebulae pass out into the large intestine. They invade the mucus lining, grow into mature trophozoites. Now, once they grow into mature trophozoites, they can either remain inside the lumen of the colon or become precystic or the minuta forms. Now, when it comes to the pathogenicity, it causes amoebic dysentery, which is around 60% of human population suffers with amoebic dysentery. It also causes problems in liver, lung, and brain abscesses it forms. And different diseases are given. Amoebic dysentery is one wherein, uh, you have passing of this like dysentery by blood, mucus along with the stools. Chronic intestinal amoebiasis is wherein you have short bouts of diarrhea with followed by periods of constipation, and there can be selective dyspepsia. That means you'll not be able to eat certain foods such as milk and greasy edibles. There's even considerable bloating sensation, then you have a flatulence, you have abdominal discomfort, nausea, nervousness, and general debility. So people with trophozoites for a long time, they'll be able to lead a normal life, and but they still are carriers of this disease. Finally, it can cause amoebiasis at any point of time when the conditions are favorable. Sometimes the trophozoites gain entrance into the circulatory system. They develop secondary extra-intestinal sites such as liver, brain, spleen, etc. This causes abscesses. There is also one cutaneous amoebiasis wherein it occurs in the perianal region or on the buttocks. So how do you control amoebiasis? Just by boiling your water, stop eating raw vegetables and fruits. Even if you would like to have salads, please wash your vegetables thoroughly. And then, you have to maintain personal hygiene. Wash your hands with soap, water before consuming a meal. And then at a community level, see that, there is no fecal contamination or fecal pollution when in the drinking water supply. So this is all about malaria. I'm going to have to stop this sharing. I'm going to share the Plasmodium life cycle if possible. So, right, screen share, advanced. So when it comes to Plasmodium, when you read the Plasmodium classification, basically you get to see that it belongs to phylum Protozoa, subphylum is Plasmodroma, class is Sporozoa, order is Plasmodidae, genus is Plasmodium, species is Wild. So one of the oldest known human diseases, everyone knows that. Greeks thought it was because of bad, damp air, so they gave the name malaria. But it's a common disease in tropical and temperate countries, and it is responsible quite, commonly for death and debility. And it's caused by an intracellular blood parasite known as Plasmodium. There are 4 species of Plasmodium present: Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae, Plasmodium ovale. So Plasmodium ovale is not exactly encountered frequently. It's there in East and West Africa and the Philippines. Plasmodium malaria or Plasmodium falciparum is the most common one, and it causes the most severe form of malaria that we know of. And for quite some time ago, till that time, what happened was they have tried to control malaria through controlling the vector, that is the mosquito. But right now you have drug resistant mosquitoes, because of which even malaria, the incidence of malaria has come up. So all the developed countries are trying to manage the vector control measures. They're trying to do that, trying to develop new drugs so that this drug resistant Plasmodium, drug resistant mosquitoes come down. so as to arrest and spread the nature of malaria. So when it comes to the people who have actually found or discovered or
అమ్మ చక్కెర
responsible
తక్కువ
for
పొద్దున్నే
people who have I request you all to please maintain silence, mute yourselves. Right. So when it comes to this, Charles Leveren was a scientist who first reported malarial parasites in the blood of a patient. Then Golgi, Camillo Golgi, that's the person's name, that's the scientist's name. He had demonstrated erythrocytic schizogony, which is also known as the Golgi cycle. And then Sir Ronald Ross, he discovered the oocysts of the Plasmodium in the stomach wall of the female Anopheles mosquito, and that was on the 29th of August in 1897. So he was working in Secunderabad, in fact, and I do not know how many of you know, but basically you have this fever hospital in near Nalakunta, Koranti Fever Hospital. It's actually known as Sir Ronald Ross Institute for Tropical Medicine or something of that sort. It's basically named after him because he was working in Secunderabad. He was working here in, the Twin Cities. So that's how it is. So,
Yes,
and he was
yes,
awarded
ma'am.
the
I know.
Nobel.
I know that
Good.
hospital.
Very nice. So that's what. So he was awarded the Nobel Prize in 1902 in the genre of medicine, in fact. So August 29th is actually known as the World Mosquito Day or the World Malaria Day, basically. So please have a look at the picture life cycle of the malarial parasite in your textbook that's given. Let me see if there is this picture anywhere. Great. So again, habitat, they have given this. I'll come back to this slide later because it's there for us and we will be looking at it. So this is the Anopheles, female Anopheles mosquito. Again, mosquitoes, there are nearly some 29 different kind of species of mosquito. Anopheles causes malaria. Culex causes some other kind of fever, viral fever. Aedes aegypti causes dengue fever. Anyway, so that's how it is. Right. This gives a very good picture of the whole, whatever is there, basically. All of it is covered in this picture. So when it comes to the life cycle of malarial parasite, you get to see that there is a sexual cycle in which you have, it is also known as the Ross cycle, if I'm not wrong. You have the ingested gametocytes, the female gametocytes, and the male gametes. You have the fertilization taking place, and over here, basically, fertilization, formation of an ookinete, oocysts beneath the stomach lining, and then you have sporogony occurring. Sporozoites develop in the oocyst, they're released and they migrate to the salivary glands. And then you have the asexual cycle, wherein injected sporozoites migrate to the liver, and then the sporozoites enter the liver cells, they undergo schizogony, and then merozoites are released. Now, merozoites enter the RBCs, they also undergo schizogony in the RBCs also. And then they form macrogametocytes and microgametocytes. And then these form the trophozoites, and then again merozoites are released. So please don't get confused, it's okay. So it's got a digenetic life cycle, as in there are two hosts. Primary host is man, intermediary or the secondary host is the female Anopheles mosquito. So transmission, as you have already known, or as you know, it's because of the bite of the mosquito. And it is similar to inoculation of the live parasite. All you can do is, take a parasite mediated, injection, in fact, and then you inoculate or give an injection. That's how parasite also can go in. Similarly, mosquito also has got the same thing, in fact, piercing and sucking mouth parts. So the life cycle of the parasite has this alternation generations and the sexual cycle, basically, which is known as the cycle of sporogony. This occurs in man, producing asexual, generation. So Asexual generation alternates with sexual generation. So this is known as the alternations of generations. So asexual phase occurs in 3 stages: pre-erythrocytic stage, exo-erythrocytic schizogony, and erythrocytic schizogony. Now, mature adult parasite is called as a trophozoite. It is amoeboid, uninucleate. Cytoplasm is granular in nature. Cell organelles like ribosomes, endoplasmic reticulum, Golgi complex, mitochondria, all of them are present. Then locomotory and contractile vacuoles are not present. Food vacuoles too are present, which may contain hemozoin, which is a decomposed component of hemoglobin. So when it comes to pre-erythrocytic cycle or schizogony, I'm trying to see if you've got a better picture anywhere, if I've got a better picture basically. Yeah, right. Good. Yes?
Ma'am, pre-exothenic exo and erythro, pre and exo difference in just
I'm coming to that. Okay, I'm just about to come to that, so you'll not have a problem with it. Look at the cycle, asexual cycle that is taking place over here. It's called as schizogony, whatever is taking place in the man. So, so pre-erythrocytic cycle starts with a sporozoite. Okay, so parasite form from the invertebrate to vertebrate host takes place in the sporozoite form. So the sporozoites come into the bloodstream of man once the mosquito bites. So sporozoite stage of the parasite along with the saliva go into the saliva, man when it bites him, and then they go into the blood. Half an hour after infection, sporozoites leave the bloodstream, enter the liver cells, especially the parenchymatous cells of the liver. They complete pre-erythrocytic schizogony. So pre. So what happens is you have pre over here. So before the erythrocytic schizogony. So in the liver cells, the sporozoites grow. They become round, called as a schizont. The schizont divides by multiple fissions. So what happens is once it divides by multiple fissions inside a liver cell, this is a liver cell. Don't get confused with the square shape. Basically, it's not the square shape that we are looking at. We are looking at the liver cell, parenchymatous cell. So in which there is this schizont which performs multiple fission. You have almost thousands of hepatic merozoites coming out. Now, they are liberated when the liver cell ruptures. This phase is called as pre-erythrocytic schizogony. This merozoites, whatever come out, they are known as cryptozoites or cryptomerozoites. Now, exo erythrocytic cycle or schizogony. Again, this is another asexual phase only, wherein what happens is these merozoites, so the cryptomerozoites of this, whatever you see over here, of the pre-erythrocytic cycle. So it is known as pre because it is before the erythrocytic cycle that is taking place. And one more thing, it is called as exoerythrocytic because it is not taking place in the liver cell anymore. These come out, they go into the schizonts, they come out as schizonts. They divide by multiple fission, producing thousands of merozoites, which are second generation. First generation was in the liver cell. Second generation, because they are from that merozoite, this metacryptomerozoites, they have come out. They are also known as, that is why they are also known as metacryptozoites or phanerozoites or metacryptomerozoites. First time schizogony is occurring. All of it is schizogony. Only thing we are naming it as pre, exo and erythro. So pre means in the liver cell. Second time in the liver cell, we are naming it as exo. So, and we are naming them as metacryptozoites or phanerozoites or metacryptomerozoites, which are again of two types: micrometacryptomerozoites, macrometacryptomerozoites. Okay, the micrometacryptozoites are more in number, and macrometacryptomerozoites are less in number. Now, this micrometacryptomerozoites, whatever are there, they go into the RBCs and they start the erythrocytic schizogony. Again, macro whatever are there, they go into the liver cell again, start again exoerythrocytic cycle. So this is what is happening. Since it is happening in the outside the liver cell, it is known as exoerythrocytic schizogony. Is it clear for you?
Yes, ma'am.
Good. Now, when we come to erythrocytic schizogony, what happens is, when the multiplication or the schizogony occurs in the erythrocytes, it is called as erythrocytic schizogony. Now, micrometacryptomerozoites, which go into the RBCs, form this erythrocytic schizogony. But some micrometacryptomerozoites get released into the liver cells for general circulation. These are the ones which actually go into the RBC to start the erythrocytic cycle. So what happens is they feed on the RBC, and then this phase of the parasite is actually known as the trophozoite phase. Let me see if I have a picture of it. I have a trophozoite picture. Right. So in trophozoite, what happens is a vacuole appears. This generally increases in size, pushing the nucleus to one side. So you get a signet ring stage. It looks like a signet ring basically. And then the cytoplasm has black pigments, which is known as hemozoin, which is a product of decomposition of hemoglobin. And then you have small granules that are appearing everywhere, which are actually known as the Schaffner's dots. And this was named after the scientist Schaffner. And then you have around it, this parasite, whatever, repeatedly divides to produce around 6 to 36 merozoites. It again differs according to the different species. And they're arranged in the, as petals of rose. So it's known as a rosette stage. I don't have picture of that basically. That is when the RBC ruptures, and then the schizons or the merozoites come out. So the erythrocytic cycle in man is known as the cycle of Golgi. All this is known as the Golgi cycle after the discoverer in the human RBCs basically. And this is taking place in the case of Plasmodium vivax from almost 48 to 72 hours. And this again differs according to the different species. Now, when it comes to the formation of gametocytes, repeated schizogony occurs, lots of parasites are formed, and then this, if you don't treat it, eventual death of the host takes place. So now what should be done to perpetuate the race? This is not going to this is not enough for the parasite. So it resorts to sexual reproduction so as to enter into the host, invertebrate host. So this is done by the adaptation of the malarial parasite which shows alternations of the asexual generations. So in man it's merozoites, whereas in the sexual generation it's the sporozoites which are present in the mosquito. So in RBCs, the merozoites grow into gametocytes. Again, there are of two types, microgametocyte and macrogametocytes. So you have two different kinds of gametocytes which are not equal. So when they, when the fertilization is occurring, it is actually anisogamy which is occurring because there are two different kinds. So microgametocyte, which is a male gametes, are small in number, and macrogametocytes, female, are larger in number. Nucleus is small, compact lysocyte. And then the mature gametocytes live only for about 1 week in the human blood. So you should, in the meanwhile, by that time itself, the female Anopheles mosquito should come and bite the man, then carry it so that, once the blood goes into the, gut of the female mosquito, that is when you have an isogamy or fertilization taking place. Again, when it comes to schizogony, there are significance of it. So you can get to see that in the pre and exo and erythrocytic, you have a huge number of offspring that is being produced. And the parasite should be able to withstand the host basically. So if the death of the host is taking place, then it means death of the parasite as well. So you have repeated asexual multiplication taking place, so that at least there is a chance for the malarial parasite to come and what do you call it bite the host. So in the invertebrate host, fertilization takes place and zygote divides again by asexual multiplication, producing many number of vertebrate hosts, parasites, I mean. So zygote generally develops into a new individual but does not reproduce asexually. So this is a unique adaptation that is there. So when it comes to the sexual cycle in Plasmodium, you get to see it's known as the cycle of Ross or Sporogony. And in this, the inactive gametocytes, the micro and the meta, after they enter into the alimentary canal, become active, develop into gametes. So microgametocytes produce male gametes, macrogametocytes produce female gametes. Then fertilization takes place. Then the nucleus of the microgametocyte actually divides to produce 6 to 8 haploid nuclei. That's by meiosis, that is reduction division. They migrate to the periphery. At the same time, cytoplasm is pushed to the surface of the gametocyte. So you have 6 to 8 flagellite-like threads. You can see this picture over here in the inbox, basically. Over here, if you see. Then these get separated from the gametocyte. Formation of the haploid male gametes is called as exflagellation or flagellation. They move into the stomach of the mosquito. Here it undergoes maturation division, develops into a haploid female gamete or the macrogamete. And then you have a fertilization cone or a cone of reception, in fact. And the microgamete gets penetrated through the fertilization cone so that syngamy occurs. Nuclei of the two, so that's what nuclei of the two gametes fuse, forming a diploid zygote with a single fusion nucleus or a synkaryon. So fusing gametes are dissimilar in nature. That is why it's known as anisogamy. After this, you have encystment, basically, in which the zygote becomes turns into the eukaryote form, basically. Over here, you can see figure 10. The zygote elongates into the eukaryote, then moves to the stomach wall. Then eukaryotes develop into the oocysts. Oocysts grow, rupture, releasing the sporozoites. Then the sporozoites migrate to the salivary glands. Then they are ready to be injected, and this renews the cycle. So this is what happens. The sporozoites are sickle-shaped, and they have a pellicle. They have hollow fibrils for locomotion. And when an infected mosquito bites a human, almost thousands of trophozoites get released into the blood. So when it comes to the incubation period, you have around 8 to 40 days, depending upon the different kinds of species of malaria that is there. So as you can see, the Plasmodium vivax causes benign tertian malaria, low death rate, but falciparum, and it stays up to 8 days in the liver and 48 hours in the, you have a table in your textbook, basically 48 hours in the blood with an incubation of 10 to 17 days. Plasmodium falciparum causes malignant tertian malaria, and Plasmodium malaria causes quaternary or quartan malaria. Ovale causes ovale malaria or mild tertian malaria. So if you come to the life history, a very good box has been given in your textbook. Please have a look at it. I do not have that picture right now, so I'm not able to share it. If you see, they have given in man the asexual, page number 28, please. In man, asexual cycle starts with the sporozoites, enters into the pre-erythrocytic cycle in the liver. So sporozoites turn into schizons, then cryptomerozoites. Then the cryptozoites enter the Exo Erythrocytic cycle into the liver. Again, schizon, metacryptozoites or phanerozoites. They are again divided into micrometacryptozoites and macrometacryptozoites. Micrometacryptozoites enter into the RBC, start new erythrocytic cycle. Macrometa cryptozoites continue with the exoerythrocytic cycle. Again, in the RBC, the erythrocytic cycle, what happens is the micrometa cryptozoites transform into trophozoites, and they have the signet ring stage and the amoeboid stage. And then this is followed by the rosette stage. And then you have the erythrocytic merozoites that are being formed. Some of them enter and become the micro and the macro gametocytes. Others continue the erythrocytic cycle. In the mosquito, again, you have a sexual cycle wherein the micro and the macrogametocytes release the gametes, micro and the macrogametes respectively. Fertilization occurs to form a zygote, which forms an ookinete to an oocyst to sporozoid, which again infects man to start the asexual cycle. So you have already seen the different kinds of malarial fever that is there. So you have three very prominent symptoms, that is intense shivering with cold, high fever and sweating. And then so you can give medicines for it, that is chincona tree is one tree wherein you have this the bark of it produces quinine medicine. So quinine is an anti-malarial drug basically, so you can administer that orally or through an injection basically. When it comes to control measures, see that hygiene is maintained, there is no stagnant water anywhere, there's an anti-malarial campaign going on because it is a global problem, and you have different kinds of control measures. First thing is you have to see that the larval forms are eradicated totally. So there are different methods to destroy the larval forms in the first place. So you can do remove the stagnant water. Then you can remove, you can add sprays of DDT, BHC, except which kill the larval forms. And you can also introduce guppy fish or minnows or trout or gambusia. So because they feed on the mosquito larvae. So you have even aquatic insectivorous plants like bladderwort, which are the natural enemies of the larva. So you have a vector control research center also that's been in Pondicherry, in fact. So they have found out a new species of water bug which actually feeds on the mosquito larvae and eggs. Citronella, it also wards off the mosquitoes, in fact. So when there are no mosquitoes, you do not have no biting, you have no malaria. That's the basic chain. When it comes to adult mosquitoes, again, mosquito repellent sprays. Then you have reproductive and genetic control, wherein they are trying to use X-rays to see that sterilized, the mosquitoes, male and female, are sterilized so that you do not have them, reproducing at all. So simple. As such, if you can protect yourself by a mosquito bite using either nets or screens or mosquito repellents, I think the problem of malaria gets stopped, because it is only through the mosquito bite that malaria is caused. So if not, all of this treatment is there. Because it changes its antigen, there is no vaccine for malaria, and they're still trying to find out a vaccine for malaria. And India is one of the countries which is at the forefront in trying to find a vaccine for malaria. So this is all about malaria. Now I will come to this last, please just a second. If you want so much of information, I can always share, but I have a feeling that you'll not have so much of time to spend only to read. So this is only for your understanding that I'm going to share this particular, PPT, if you would like to have, that is. Please give me a moment. Let me see. I have another PowerPoint over here. Right. So the last topic, or the last part in our this particular unit is Trypanosoma Gambiancy. It causes sleeping sickness, or the African sleeping sickness. It's a hemoflagellate, again parasite in blood, causes trypanosomiasis, and then it's about 20 species of trypanosoma formed. But then only Trypanosoma cruzi and Trypanosoma brucey form cause diseases in humans. and each species is responsible for a different disease as such. This is known as Chagas disease and Trypanosoma cruzi, that is transmitted by Tryatoma, and then it is also known as spread by the bite of insects, which are known as the kissing bugs. Trypanosoma brucei and Trypanosoma, subspecies is Gambiancy, because it is found in Gambia, Rhodesiense. It causes African sleeping sickness. It's transmitted by Glossina palpalis, which is the tsetse fly. So the West Africa, and it is an acute form. Disease symptoms occur in a very short span of time. So the tsetse fly, both these forms are actually transmitted by the tsetse fly, and it causes several other important diseases by other species of the Trypanosoma also. So you have cattle also having sleeping sickness. So it causes an economic loss, in fact. So this chapter has us describing about Trypanosoma brucei Gambiancy. So we are going to see about this. So it was discovered by a scientist known as, this is how, they start sleeping. So you have the West African or Gambian, and this is the Rhodesian, East African form. So, right. So it was discovered by David Bruce. That's why they gave the name David Brucey, in fact. And he had suggested, because the Nagana cattle in Zululand, the Zululand is in Africa, so it caused the sleeping sickness in the cattle, in fact. And then Ford and Dutton reported that the parasite in the human beings in Gambia, that is Africa, and Stephens and Fathom in 1909 in Rhodesia. So Klein is one scientist who had asserted that the intermediate host in this life history was a tsetse fly. So Trypanosoma species in their life cycle exhibit different morphological forms. Therefore, it's a polymorphic form. So you have, based on the position of the parabasal body, the basal granule, and the course of the flagellum. Some polymorphic forms are amastigophore. So it's known as a this one. Amastigote, leishmanial form, body is round or oval with a nucleus. Basal granule, parabasal body are completely reduced. And at the anterior end, free flagellum flagellum is reduced, does not extend out of the body. And this is the promastigote, which is known as the leptomonad form. So you have a body which is elongated with a central nucleus. Basal and parabasal body are present at the anterior end, which is anterior to the nucleus. Flagellum arising from the basal granule, and then there is no undulating membrane present. Then this is the criteria or the epimastigote form. So body is short, elongate but stumpy. Nucleus is central. Basal granule and parabasal body are just in front of the nucleus. Flagellum arising from the basal granule is at the anterior end, and it is free in nature, as you can see. Undulating membrane is present, but it is very poorly developed. And this is the trypanosome form, which is known as the trypomastigote. Body is elongated, slender. Basal, parabasal body are present at the posterior end, as you can see over here. Flagellum is free after emerging, and you have an undulating membrane which is very well developed. And you also have a very good picture of the same in your textbook, so you can just have a look at it. And when it comes to the habits and the habitats, it's a plasmodium parasite present in the blood, lymph, and cerebrospinal fluid of man, and it goes into the regional lymph nodes also. And then it invades the bloodstream, causing parasitemia. And then finally, its total or final destination, as you call it, is the brain. So it causes African sleeping sickness, and this is a disease of the central nervous system. So when it comes to the morphology of the insect, you have Trypanosoma brucei gambiancy exists in the trypomastigote form in the human beings. As you can see, it's with the undulating membrane and all that. So anterior and posterior, anterior end is pointed, posterior end is blunt. 60 microns length, 15 microns width, and body is with a strong elastic cuticular pellicle. Mouth is not present. Cytoplasm is divided into ecto and endoplasm. Body is thin, lateral fin fold, causing undulating membrane. Nucleus is large, round, disc-shaped, and then you have a thin. Then towards the posterior end, you have a blepharoplast and the parabasal body, in fact, and then you have the parabasal body or the kinetoplast present. Flagellum arises from this, what do you call it, basal granule, travels along the margin of the undulating membrane, then comes out as a free flagellum at the anterior end. In the cytoplasm, you have something called as greenish refractile granules, which are known as volutin granules. So this exhibits polymorphism again. Parasites vary in shape and size in the different stages of their existence. So you have long, slender with a long free flagellum, short, thick, stubby form with no or less flagellum, intermediate forms. Again, it's a digenetic parasite. You have two different hosts that are present. So primary vertebrate host and the secondary invertebrate host. Primary is man, or you can have game animals such as deer or etc. And then you have domestic animals such as cattle, which are infected. And the invertebrate host is the tsetse fly, which is the blood-sucking fly, basically, Glossina paparis. So parasite multiplies by asexual binary fission. We do not know about the sexual fission or reproduction in the, what do you call it, in this particular protozoan. So when it comes to development in man, this infected Glossina, what do you call it, bites the man, inoculates a few parasites into the blood. These are called as the metacyclic trypanomastigotes. They transform into the bloodstream, into the bloodstream trypanomastigotes. Then they are carried to the other sites. They multiply by binary fissions in different body fluids, blood, cerebrospinal fluid, lymph, etc. Then this is how it is there in the blood, basically. Then when if it is there in the blood, that is when the tsetse fly bites, sucks this trypomastigotes. This goes into the bloodstream, into the procyclic trypomastigotes in the tsetse fly's midgut. Forms a procyclic trypomastigotes, multiplies by binary fission, and then it forms epimastigotes, which multiply in the salivary glands, transform into the metacyclic trypomastigotes, which gets again injected when this tsetse fly bites man. It gets injected over here. So it's the metacyclic trypomastigotes which get injected into the man. So this is how it takes place. And unlike the malarial, so what happens is when it goes into the cerebrospinal fluid, the lymph, etc., they form, like, what do you call it, they reach the cerebrospinal fluid, they disturb the central nervous system, they cause the sleeping sickness. So unlike the malarial parasites, Trypanosoma does not enter into the RBCs, but it remains outside in the plasma of the RBC, outside the, so it's in the lymphatic system basically and the plasma system. So the parasites in the blood, the host develops antibodies against the parasites. These antibodies kill most of the parasites. So what is happening is, since it is there in the plasma, it is evading the host defense system, again by changing the antigens on its body surface. So this is the most important parasitic adaptation which is seen in Trypanosoma Gambiancy, because of which we do not have any kind of vaccine that is being produced against Trypanosoma Gambiancy. Now, when it comes to the development in tsetse fly, as I told you, it enters into the through the blood into the midgut of the fly. Short stumpy forms, whatever are there, all the trypomastigotes only, basically, they multiply by longitudinal binary fission. In 10 to 15 days, the long slender forms appear in large numbers. They move to the proventriculus, where again they start multiplying, and then again through they make their way to the salivary glands of the tsetse fly, and over here they develop by longitudinal binary fission into the criteria forms. Later these develop into the metacyclic or the trypanosome forms, which are infective to man. So this again enters into the blood of the man through the saliva of the fly. So the whole life cycle in the fly with the evolution of the infective stage takes about 20 days. Infected stacey fly remains ineffective for a period up to 185 days, after which it can cause infection, and that is very easily observed with the symptoms appearing. So when it comes to the pathogenicity, you get to see that symptoms of sleeping sickness, you after the first basic bite, it can be after the symptoms you can see after quite some time basically. So you have fever, which is there after maybe it can be after 2 weeks of the bite also. 3 stages into the disease that is there. It's followed by local itching, irritation. Fever is intermittent. You have button-like lesions that are formed, hard, painful nodules. This subsides in a week or two without any kind of pus release. Suppuration means release of the pus. Then after a few days, when the number of parasites increases in the blood, high fever occurs at regular intervals. Then you have headache, anemia, you have acute weakness, enlarged glands, you have edema, that is, swelling everywhere. And then you have low resistance is the scene. Your immunity system has come down basically. Because see, you're producing antibodies against certain known antigens, but it is again changing its antigens, because of which you have fever being produced, you have weakness, all that is associated to try to kill the parasite, but that is not happening because it is evading the host immune system. So when the parasites invade the cerebrospinal fluid of the brain, the spinal cord, then that is when the patient starts getting disturbed. There is a feeling of oppression. He or she loses consciousness. Finally, they go into the comatose or confusion stage, and then they become comatose, that is, go into coma, and this ends in death. So do you have any medicines against this? Let us see. So see, that's what antibodies produced in the first week against clone A will not react with clone B. So changing display is called as antigenic variation, which is an important form of immune variation. So again, so what are the preventive measures? So when it comes to the preventive measures, destroy the habitat of the vector, which is the tsetse fly. So this is done by clearing the bushes and low trees along with the rivers in the endemic areas, and then eliminate the fly by using insecticides, elimination of the reservoir in the affected parts of the country, and then you isolate the people who have been, affected with this from the endemic areas and treat them first. You have certain medicines, intramuscular and intravenous, pentamidine or suramine for that matter. Try to keep giving them that. Let us see, that's what. But then, as I said, the first three are quite disadvantages, because destruction of a habitat, again, it's going to be having a huge impact, negative impact on the environment, and the animals that live in that areas. So you can spray insecticides, but again, it's not very practical in very large areas, as the insecticides are not exactly specific to a particular species as such. So it is not beneficial to man also. So what happens is killing of game animals, that is deer or antelopes. Again, if you do that, what will happen? Because they are vectors, extinction of these animals. So there is not exactly any kind of foolproof control of this parasite. But fortunately or unfortunately, trypanosoma is one thing which is endemic or localized or mostly seen in Africa only. So this is all about Trypanosoma Gambiancy, that is the African sleeping sickness. So the next class, I'll try to deal with Porifera and if possible even a little bit of Nidaria. We'll have to be I'll have to be able to tell you a little more. So I'll just, go ahead with the general characters and the classification and the type study. Canal system, just touch them all once and then go ahead with the Coelenterata, because that should also have been completed by tomorrow, in fact. So this is all that is there for today's class. You can all leave right now. I'm ending the class. and then come back tomorrow again. Yeah, thank you again.
Thank you.
Thank you, ma'am.
Thank you, ma'am.
If you want, I will, if you want, I will share this particular slide with you, but if you feel that, you know, you do
Thank you,
not have the time, yes.
Thank you, ma'am.
You want me to share it or not? Because I feel, you know, whatever is there in your book, if you read that itself, you know, is like more. And you've got better diagrams and, you know, better pictures that have, you know, much more easier for you to understand in your textbooks, basically.
Okay, ma'am. Madam,
Thank you,
please,
ma'am.
please share me that PPT.
Wanted some important questions.
Important question.
Important questions that were just like, you know, see what is there inside the, like, you know, model questions. I think you have model question papers right behind, you know, your textbook. You can see there is one model question paper, zoology at least. You can see.
Ma'am,
Every page of your textbook, you have one model question paper. You can just go ahead. In case you want any of them, a little more of them, then you can, you know, you can try to contact Kishore sir. He will be able to help you. Okay.
Okay, ma'am.
All right.
ma'am.
All right. Then I will be sharing this
Okay,
PPT, but it is only for you to understand or help and look at the like, you know, PPT. That's all. It is not going to be of much use to you. Please follow the textbook religiously. Okay.
Okay, ma'am.
All right. Then thank you so much.
Hello,
You can all.
hello, madam.
Yes.
Hello,
Yes.
madam.
Chapandi. Yes.
Can you share me that PPTs? Can you share
I
me that
think you
PPTs
have.
and important notes like that?
Yes, you have a group, right? Like I will that post the PPTs in it. But unfortunately, if you know
Yeah,
anybody
in that.
from the Telugu medium, in case they would like to have, you can pass it on to them also. Okay. If you know, if you happen to know anybody from the Telugu medium also. Okay. All right, then. Thank you so much. You can leave now for the day.
Thank you, ma'am. Thank you, ma'am.
Thank you.
Thank you.
Thank you, ma'am.
Thank you, ma'am. Thank you.
Srikanth.
Hello. Chapandi.
Okay. Okay. 31 members is there. Can I talk with you individually?
Yeah, okay, sure.
Jyoti.